Okay, welcome to this video on the lymphatic system and immunity. First, we need to know that the lymphatic system includes the cells, tissues, and organs responsible for protecting the body against environmental hazards and internal threats. And the lymphatic or immune system, depending on which book you have, it's called both, does the same thing. It is made up of, first of all, the lymph vessels, which are these green vessels we see here. The lymph nodes, this is a lymph node, and they're found all along the lymph vessels. We also have the tonsils around your mouth, and here's a close-up on one. Peyer's patches, which are found in the intestines and along the reproductive tracts. This organ here is the thymus. And the spleen. The spleen is the largest lymph organ, and that's where the spleen is located in your body, next to your stomach there. And then also the immune cells, the white blood cells. So we're going to review something we talked about last time with filtration of blood capillaries. We can see that at this end we have 24 liters a day of fluid leaving and only 20.4 liters entering. So that other 3.6 liters, today we're going to answer the question of what happens to it. And it goes into the lymphatic system.
We have learned that the immune system cells and the lymphoid organs and tissues are the components of the immune system. The lymphatic system overlaps the immune system in structure and function. We will now briefly look at the lymphatic system. The lymphatic system has three parts: a one-way system of vessels called lymphatic vessels, the fluid in those vessels called lymph, and the lymph nodes. Click a lymphatic vessel to begin. The smallest lymph vessels, the lymph capillaries, weave through the blood capillary beds of loose connective tissue. They are blind sacs or closed tubes formed by overlapping endothelial cells. The overlapping endothelial cells are supported by collagen filaments that anchor the cells to the surrounding matrix. As fluid builds up in the tissue, the overlapping flaps of the endothelial cells open and the fluid enters the capillary. These overlapping endothelial cells act as one-way valves, allowing fluid to enter but not leave the lymph capillary. Click the lymph capillary to see. Plasma proteins, pathogens, antigens, and dendritic cells can also enter the lymphatics from the tissues and be transported to nearby lymph nodes. Click the blood capillary to see lymph flow through the tissue.
So you can see those bad things, those bacteria, they were taken.
Capillary to repeat the animation. Click the lymphatic capillary to continue. Lymph capillaries lead into larger lymph vessels, which, like veins, contain valves to ensure one-way flow of fluid. Interspersed between segments of these larger lymph vessels are the lymph nodes. Lymph filters through the nodes and is cleansed of antigens and pathogens by phagocytes. Lymph is funneled into successively larger lymphatic vessels until it finally drains into the circulatory system and the large neck veins.
Okay, so as we saw in that video, not all the fluid that leaves a capillary goes back into a capillary. Some of it goes into the lymphatic system, and it takes with it bacteria and other bad things. So the lymphatic system takes this fluid, cleans it, and returns it to the blood. Now the lymphatic capillaries are associated with capillary beds throughout the body. Just like this one, here's the capillary bed, and the lymphatic capillary is right next to it. If we look closer at one, we'll see these endothelial cells overlap to form one-way valves, so those will let things in, so water and any pathogens that it might bring with it enter the lymphatic system. And these are very similar to blood capillaries, except for they have thinner walls, they have a much smaller diameter, they originate as pockets. Blood capillaries are continuous, these start out as pockets. And there's less pressure, much less pressure in the lymphatic system. And as we saw in the video, the lymphatic capillaries will join together to form larger lymph vessels which have valves. Here's a diagram of a valve right there. And here's an actual lymph capillary, and we can see the valve. And they're just like the valves in veins that keep the fluid flowing one direction. Now these small lymphatic vessels converge to form lymphatic ducts which empty into these subclavian veins. As we can see in this picture, here's the ducts, and they drain right there into the subclavian and also right there. This is what happens when the lymph drainage is blocked. It's called lymphedema or elephantitis. Pretty uncommon in the US. Next, we're going to talk about the white blood cells. So these are lymphocytes. About 80% of them are T cells, and we'll learn why they're called T cells in a minute. The first type of T cells are the cytotoxic or the killer T cells. I like calling them killer T cells, that just sounds cooler. These are the cells that can directly attack and destroy foreign or infected cells, and they are responsible for cell-mediated immunity. And we'll learn all the details of cell-mediated immunity later on in this movie. We also produce helper T cells. These are going to stimulate the activation and function of T and B cells, so we need these to start the immune response. And then there are also suppressor T cells, which inhibit the T cells and the B cells. We don't want our immune system to go overboard. That's what leads to autoimmune diseases, so these cells are important as well. Then about 15% of the lymphocytes are going to become B cells. B cells differentiate into what are called plasma cells. These are the cells that are going to produce antibodies, and we'll learn about that process later. And these cells are also responsible for what's called antibody-mediated immunity. And then the other 5% of lymphocytes are going to become what we call natural killer cells. These attack foreign, infected, or cancer cells, and they're responsible for immunological surveillance.
Of all the immune system soldiers, natural killer cells are the most aggressive. They are lymphocytes with no immunological memory and are part of your innate immune system. Natural killer cells make up about 5 to 15% of the total circulating lymphocyte population. Natural killer cells are called natural killers because they do not need to recognize a specific antigen before releasing their toxins and destroying a viral infected cell or cancer cell.
That green thing is a virus.
And protect against a wide variety of infectious microbes. In several immunodeficiency diseases, including AIDS, natural killer cell function is abnormal, thus allowing the viruses to multiply inside of human cells.
So you have an infected cell there in the middle.
Kill on contact, the killer cell binds to its target, aims its weapons, and then delivers a lethal burst of toxins that produces holes in the target cell's membrane and creates events that destroy the cancer cell membrane and cleaves DNA into many pieces in a programmed cell death, that is, apoptosis manner. After releasing its lethal package of chemicals, the natural killer cell can detach from the cancer cell and kill many other circulating abnormal cells. Indeed, research has shown that most of the tumor cells that enter the circulation are
Okay, so that's a diagram of the natural killer cells, and here's an actual picture of one. There's a natural killer cell, and you're going to see it release its toxins, and you're going to see eventually cell death. So this really does happen, we're not just making it up. So that top cell was something foreign or something infected, and the natural killer cell is killing it. And now it's going to move on and kill something else. Okay, lymphopoiesis, or the production of white blood cells, occurs in bone marrow. And so all the cells start off in the bone marrow. All of our blood cells, including white blood cells, are produced in the bone marrow. Some cells are going to go to the thymus to mature, and they become T cells. The rest mature in the bone marrow, and they're going to become B cells, or they could become natural killer cells. Once they're formed, lymphocytes hang out in the lymphoid tissues and organs, such as in the malt. The lymphoid tissues that protect the digestive, respiratory, urinary, and reproductive tracts form what's called mucosa-associated lymphoid tissue, or MALT. And here's an example of some from our intestines. Tonsils are the large lymphoid nodules guarding the mouth and nose, so anything that we eat or breathe in. We have this ring of tonsils around our throat guarding it. And here's a close-up on the tonsils showing the large crypts. And in those crypts we have lots of these white blood cells, and they'll destroy anything. Unfortunately, sometimes those get infected, and that's when people have to have their tonsils removed. So the lymph vessels lead to lymph nodes. So here's some lymph nodes, and here's a closer up look at a lymph node. And you can see the lymph would be coming in from these four, and actually it would be more than that. These lymph nodes are going to filter the lymph. So here is an even closer look at a lymph node, and you can see it's made of this reticular tissue, which sort of looks like a filter, and it acts like a filter. And so these cells will destroy anything foreign, and they will also activate the immune response if they find something foreign. Okay, the thymus produces functional T cells. The thymus is right here. The thymus produces hormones called thymosins, which promote the development and maturation of lymphocytes. And T lymphocytes become what we call educated in the thymus. So the first step is called positive selection. And cells that don't recognize our self-MHC proteins are destroyed. And cells that recognize our self-antigens too strongly are eliminated, because those might cause an autoimmune problem. They would attack our own cells. So the cells first have to be able to recognize our self-antigens, but not too closely. So that second part is negative selection. If it recognizes our own cells too strongly, it will be eliminated. But if it recognizes it weakly, then it continues to survive and mature. And after puberty, the thymus becomes really fibrous and shrinks, so we only see it in children. Next, we're going to talk about the spleen. The spleen is the largest lymph organ, and we're going to watch this video to learn what the spleen does. if you want to see that video again, if you didn't catch what the spleen does, you can find that video on YouTube. So if we look at the spleen, it's full of lymphocytes, macrophages, and dendritic cells, and there's basically two parts to it. The red pulp, which looks red, and the white pulp, which looks white. The white pulp is full of white blood cells, and it's going to destroy any foreign objects like a bacteria or virus that got into the blood. And in the red pulp, our worn out red blood cells and platelets are destroyed by macrophages. So you remember, red blood cells don't live very long, only 3 or 4 months, and so when they're no longer working, the spleen takes out the old ones. Alright, so now we're going to cover the nonspecific defenses, or the innate immune system, and this is a summary of them. So first of all, you should know that an antigen, when you hear the word antigen, that could be a pathogen, any foreign object like a bacteria or a virus, an infection, an invader, or a bad thing. We call that an antigen, and it's going to activate the immune system. So let's look at these non-specific defenses. So these will kill anything foreign. So the first one is physical barriers, such as the skin. And then we have phagocytes. These are cells that will find a bacteria and swallow it and destroy it. We have immunological surveillance, which is done by the natural killer cells that goes around and looks for anything foreign and destroys it. We have interferons, which are chemical messengers that can destroy foreign things. We have the complement proteins. It's a system of proteins that our body uses that helps the immune system to destroy pathogens. We have the inflammatory response, which helps to limit the spread of an injury or infection. And we have fever. Fever's elevation of body temperature. It's going to increase your metabolism, and it's going to kill some bacteria. So a slight fever is actually a good thing when you get sick. But if it gets above 104, then it might cause brain damage, so you don't want that. Alright, so first we have physical barriers. They keep bad things out. And our skin is the first line of defense. Our hair is also part of this. The hair protects the skin from abrasion, and it helps keep things off of the skin. And as we see here, skin has several layers of keratinized cells. These keratinized cells, they're tightly connected by these desmosomes. And you learned about how the skin forms, about the layers of the skin last semester. So these are the mature keratinocytes. You also have the sweat and oil glands. They secrete bactericidal chemicals, and just having sweat on there, that could knock off any bacteria or bad things.
Mucous membranes line all body. Skin acts as a physical barrier to infection. 3 properties of the skin are key to its ability to resist invasion. First, the outermost layer contains the tough protein keratin, forming an abrasion and water-resistant surface. Second, specialized intercellular junctions, such as desmosomes and tight junctions, hold skin cells tightly together. Third, skin secretions are acidic and contain chemicals that make the skin an inhospitable place for pathogens. One of these secretions is the enzyme lysozyme, which can digest the cell walls of certain bacteria. Click the skin.
So you can see the pathogens just bounce right off of the skin. Or the sweat might kill the pathogens.
Mucous membranes line all body cavities that are open to the click the mucous membrane. Generally, insert click the trachea. Pathogens and other particles suspended in the air, such as dust and pollen, enter through the nasal cavity. All but the very smallest of these are trapped in the click the cilia to see them
So here we can see bacteria getting trapped in the mucous membrane, and then as the cilia move, they move it up and away from the lungs. So that's how our mucous membranes work to protect us. So just as we saw in that movie, here's a mucous membrane, and pathogens get trapped in it, and the cilia will move it away and out of our body. So mucous membranes trap and kill foreign objects. If something manages to get through the skin and the mucous membranes, then it would next encounter the phagocytes. Phagocytes provide the next line of defense. Neutrophils, eosinophils, and monocytes all form phagocytes. Some phagocytes are fixed in a particular tissue, like a lymph node. There's lots of lymph nodes. There might be some in your lungs. Some are free to roam around. Here's a picture of one. In yellow is the phagocyte, and the orange shaped thing is a bacteria, the orange colored thing. Here's a video.
Neutrophils are white blood cells that hunt and kill bacteria.
Of one that can roam.
A neutrophil is seen in the midst of red blood cells. Staphylococcus aureus bacteria have been added. The small clump of bacteria release a chemoattractant that is sensed by the neutrophil. The neutrophil becomes polarized and starts chasing the bacteria. The bacteria, bounced around by thermal energy, move in a random path, seeming to avoid their predator. Eventually, the neutrophil catches up with the bacteria and engulfs them by phagocytosis.
Okay, now that we've seen a phagocyte in action, we need to discuss their properties or their characteristics. So some important things they have is that they can actually leave capillaries, and they do this by squeezing through the cells, and that's known as emigration or diapedesis. So diapedesis is one characteristic, and that's what allows them to leave blood capillaries, and that way they can go out into the tissues wherever the bacteria or whatever is and destroy them. Another important property is called chemotaxis. They're attracted or repelled by chemicals in the body. So if there's an injury, we're going to release chemicals that will attract phagocytes to the area. Phagocytosis begins with the phagocyte attaching to its target. The phagocyte may destroy the target itself, or it may activate other defenses to destroy it. So next we have some review questions. Most pathogens that try to enter our body are stopped by. And the correct answer is C, the skin and mucous membranes. The primary function of the lymph system is... Correct answer is A, defending the body against external hazards and internal threats. Alright, next we're going to talk about immunological surveillance. So natural killer cells respond to anything foreign. So anything that's not part of your body. Anything foreign, infected, or cancerous. And the natural killer cell will stick to its target, and then you can actually watch the Golgi apparatus move. It will move around. And it makes perforins. Perforins are proteins that are going to punch holes in the target cell membrane. These perforins are being released here. And they form holes in the target cells. So if you look at this close up, there's the holes being formed by the perforins and the cell lyses. So when the hole forms, water is going to rush into that cell and cause it to basically explode. And that's what you see happening here. So as cells divide, sometimes something happens. There might be a problem with transcription or problem with copying the DNA and you get a cancerous cell, or maybe the cell gets infected by a virus or something. And most of these abnormal cells are going to be recognized by the natural killer cells and killed. So here it is, killing that cell. If they're not recognized by the natural killer cells, then they might become cancerous cells. Well, they were already cancerous cells. But if a cancer cell escapes detection by the natural killer cells, it may form what's called a secondary tumor. So here's the cancer cells. Most of them got destroyed by natural killer cells, but this one got away, and it's forming a secondary tumor. This is known as metastasis. Alright, next we need to talk about interferons. Interferons are released by activated lymphocytes and macrophages, and by cells infected with viruses. So here's a cell with a viral infection. It's going to release interferons. They can also be released by fibroblasts to slow inflammation, and they're also released by T cells and natural killer cells to stimulate macrophage activity. When it's a virus infected cell, what happens is once a cell's infected by a virus, it starts to make interferons, and then those interferons will be released and go to neighboring cells, and they make antiviral proteins which interfere with viral reproduction. And if that virus can't reproduce, then it won't work in that new cell. So this cell basically has to give itself up. It knows it's infected with the virus, but it makes the interferons to protect the surrounding cells. And interferons also stimulate macrophages and natural killer cells. Next thing we need to talk about is complement. There are 11 proteins circulating in the blood that make up the complement system. In the classical pathway, which we see right here, in the classical pathway, antibodies bind to a foreign cell, and when one complement protein binds to the antibody, it attracts other proteins until an activated complement protein is formed, and then they're going to form what's called a membrane attack complex and form a pore in that cell. There's also the alternative pathway, which does not require antibodies. Complement proteins can interact in the blood plasma if they're triggered by exposure to foreign materials, and they will form an activated complement protein. And this activated complement protein will form pores in the target cell wall. It also causes what's called opsonization, which makes them more attractive to macrophages. So if a macrophage sees a cell with complement on it, it's going to know to destroy that cell. And it can also release histamine to increase blood flow to that region, and with that increased blood flow will come more of the white blood cells, more phagocytes, to hopefully take care of it. Okay, next we're going to talk about inflammation. Inflammation is a localized tissue response to injury. It causes heat, redness, swelling, and pain, all due to the increased blood flow in the area. And what causes inflammation is the mast cells releasing histamine. This is going to dilate the blood vessels and increase their permeability, so more blood is going to come, and also more blood is going to leave the blood vessels. Histamine also attracts phagocytes. That's known as chemotaxis. So it's going to attract the phagocytes to the region. When those phagocytes come, they're going to release cytokines. This leads to debris removal and activation of the specific defense system, which we'll talk about next. Another non-specific thing is fever. Fever is caused by pyrogens, And that increased body temperature will kill some bacteria, and it will increase your metabolism so that you're better able to deal with fighting off the infection that you have. So, a summary of the non-specific defenses. The first line of defense a pathogen is going to encounter would be the physical barriers, the skin and mucous membranes. If it gets past that, the phagocytes might destroy it, or immunological surveillance, the natural killer cells might find it and destroy it. Interferons can also help destroy things, and they will attract these phagocytes and natural killer cells. And then we also have the complement system that can destroy things, the inflammatory response due to the mast cells, and fever. Those are all the non-specific defense mechanisms that our body uses. Okay, so the part of the innate defense system responsible for immunological surveillance is, and the correct answer is natural killer cells. The classical pathway of complement activation begins when the complement protein binds to, and the classical pathway involves antibodies, so the correct answer is D, an antibody attached to an antigen. An inflammatory response is triggered when mast cells release histamine. The part of the innate defense system activated by pyrogens is, and the correct answer is fever. Pyrogens, like a pyromaniac makes fire. Pyrogens make you hot like fire. Okay, so specific or acquired immunity can be either active or passive, and it can be either naturally or artificially acquired. So passive, naturally acquired, you don't have to do anything. This would be like babies can receive antibodies via the placenta or through breast milk. Then it can be passive, artificially acquired. This is when antibodies from an outside source are injected. So this would be like if somebody gave you anti-venom for a snake bite or something. That would be passive, artificially acquired. And then active immunity, to get that your body has to do something. So active, naturally acquired, develops after exposure to antigens in the environment. So for this one, you have to get sick. It's not fun. Nobody likes to be sick, but it does build your immune system, and this is the best way to build it. Then we have active, artificially acquired, and this would be like a vaccine. When they give you a vaccine, they're giving you a weakened or a dead virus, and so it's not going to make you sick, but your body recognizes the antigenic sites on it and can develop antibodies to it. So that way, if you do encounter that pathogen, then you have the antigens for it already, or you have the antibodies for it already, and you can destroy it right away. Alright, next we're going to talk about some properties of immunity, and we're going to focus now on the specific defense system. So we have what's called specificity. So the T and B cells are going to be specific for one antigen, such as chickenpox. And other properties are versatility. So we have millions of different lymphocytes, and they're specific for millions of antigens. So we can respond to a wide range of infections. A lot of pathogens that enter our body are actually destroyed by our immune system before we even get sick. Another characteristic is immunological memory. Memory cells enable the immune system to mount a stronger, faster, longer lasting attack if you encounter the same antigen again. That's why you usually don't get sick with the same thing twice. And another important property of the immune system is tolerance. That means the immune system it ignores our normal cells, so we don't destroy our good cells.